Hey there, everyone. We'll go ahead and let folks start coming into the Zoom room. In the meantime, go ahead and use the chat window at the bottom of your Zoom screen. I would love to hear from you. Go ahead and let me know if this is your first Vernier webinar or if you've joined us for a webinar, before or not. I'd love to know who's new. Oh, looks like Allison's first time joining us, and Charlie. Hey everybody, nice to see you all. And Kate, thanks for letting us know. All right, well, as people kind of keep coming in the room, I will pass off the mic to Melville as it is here in just a second, but I did want to do just a little housekeeping. You'll be hearing from me throughout the webinar. I have a poll to give you all before we dive in. I also will be giving you kind of a survey at the end of the webinar. I've got lots of resources, so I'll be dropping a link to our resource folder in the chats. In there, you'll be able to see the presentation Melville's going over, so you can click the links that are in there. You'll also see the certificate of attendance that you get for attending this webinar, as well as some experiments that you can go ahead and do in your class. Along with that, we will be recording this webinar today, so you'll be able to watch the webinar on YouTube as well as on our website as soon as that's live. It usually takes us about 3 days to a week, so keep an eye on your inbox. I'll email you as soon as that's live. I am more than happy to answer your questions, so drop your questions in the chat and I'll answer where I can. If there's something that I can't answer, I'll stop Melville and I'll ask your question to him. So please feel free to stop us, let us know what you're wondering, and we'll go from there. And with all that being said, I'm going to go ahead and mute myself and I'm going to start that pre-webinar poll. So keep an eye on your Zoom screens, everyone. That's coming your way.
Yeah, it's just a really simple poll just to ask a few questions. You know, basically, are you a Project Lead the Way educator? And then are you here to learn how to use our new Go Direct sensors, or are you more interested in the old ones, just to get a feeling? Well, I'm going to cover both. And anything that we do cover in this webinar, remember, it's going to be recorded. We have lots of other resources for you. And if there's something that I don't cover, you can always contact me at biology@vernier.com and ask for a one-on-one training. As long as it's under an hour, it won't cost anything. And if there's enough people that want us to go over something that I haven't covered in this webinar, we can do another one. All right, so go ahead and take some time to answer that, and then I will let Dylan tell me the results of the poll, and then we'll get started. Great, so it's about half and half. Okay, great. And oh, great, it's about half and a half. That's what I thought. So some people are here, some people are Project Lead the Way teachers, some are not. Some are here for Go Direct sensors and some are for old. So don't worry, we'll go through it all. You don't have to be a Project Lead the Way teacher to understand how to use human physiology sensors and the ones that we're going over. All right. So let me what we're going to be basically doing today is the agenda is you're going to meet your presenter, which is me. My name is John Melville. Everybody calls me Melville, as you just saw Dylan refer to me as. I'm going to be focusing mostly on the Go Direct EKG sensor, and then also the EKG sensor, which is older, and show you how they work and how to set them up. But I'm also going to be talking about our other human physiology sensors, both old and new, and show you how you can use them in graphical analysis or Graphical Analysis Pro. Graphical Analysis Pro is our new software that can be used on Chromebooks, iOS devices, Android tablets, and Mac and PC. So it's really useful. It can be used in anything. For a lot of people that are used on Chromebooks, the UI, or what we call the user interface, is the same. So that when I'm showing you graphical analysis, it's going to look exactly like it would on a Chromebook. Don't forget, you can ask questions anytime in chat. And I'll try to make sure there's some time for Q&A or questions and answers. All right. Great. So just a little bit about me. That young guy over there was me. I have a BA in biology and psychology from Sonoma State University. I got my PhD in zoology from Oregon State University. I did postdoctoral research at the University of Oregon in neuroscience, but most importantly, I was an assistant professor of biology at a place called Wartburg College. And I used to teach at a Lane Community College as well. But my real focus in both places was teaching majors and non-majors courses in biology, neuroscience, and I had a real emphasis in active learning with an emphasis on data collection. So we did a lot of human physiology in our intro bio course. And also at Oregon State University, I was one of the first people to actually integrate data collection technology in the animal physiology courses, and actually also worked with the human physiology folks there. So I have a long history in data collection. And you could just call me Melville. It's, my name is John Melville. I'm the director of the biology department and the biology staff scientist, but most everybody calls me Melville. All right, so just don't forget, you can ask all your questions in chat. Someone will be there to answer your questions as we go. If time allows, we'll discuss questions at the end of the talk. If you don't already have graphical analysis, you can find it on your device, download it if you don't already have it. You can get it on our website. I can show you how to get it. If you go to vernier.com, if you just tap on the top right where it says downloads, it'll take you to graphical analysis, and you can go and you can download it. And the base graphical analysis is free. You can totally download it. You don't need to do anything special to use it. There's Windows, Mac, Chrome, iOS, and Android versions. We also have a really cool, what we call a progressive web app. So if you have if you have Chrome and you have an open browser, you can type in graphical analysis. .app, and it will launch graphical analysis. All right, so someone had a question that looks like there's a graphical analysis GW and a different graphical analysis. Graphical analysis GW stands for Go Wireless, and that is the old version of graphical analysis. We're using real graphical analysis, so don't use graphical analysis GW. You want to use normal graphical analysis. Don't worry, how does graphical analysis compare to Logger Pro? We'll get to that. Logger Pro, remember, can't be used on iOS or Android or Chromebooks. Graphical analysis can, so that's the biggest difference. And the UI is updated. There's this really cool GA Pro key that you can use. And that's really useful. If you launch graphical analysis, you can put that down here at the bottom. You can put in that code. Once again, the code is FZK91VPYI. I'm going to put that in the chat in case everybody wants. F. Just give me a minute. FZK91VPYI. And so when you launch graphical analysis, you can put that down here in the bottom and activate it. And that'll give you what we call all the pro features. And here's the pro key again. All right. But off to what we're really talking about. Oh, the other big thing, the important thing between Logger Pro and graphical analysis is that these are our new sensors. This is a Go Direct EKG, Go Direct Hand Dynamometer. This is a force sensor with a reflex hammer attached to it. The Go Direct sensors cannot be used with Logger Pro. So all of our new Go Direct sensors cannot be used with Logger Pro, but all of your old sensors that you have can be used with graphical analysis. And that was one of the things that I was going to show you. All right, so what I want to talk about first is how great our Go Direct EKG sensor is and how to use it in graphical analysis, because lots of people want to look at EKGs, and then talk about some of the things that it can be used for. You can study EKGs with it, EMGs, which is electrical activity of muscles. You can look at reflex activity, and it also can measure heart rate. Most of what I'm going to be showing you are based out of two books. We have our Human Physiology Experiments, Volume 1, and then we have Human Physiology Experiments, Volume 2. Volume 1 is really designed more for intro level, high school. It uses fewer sensors, EKG, heart rate, etc. Human Physiology Experiments, Volume 2 is the one that actually uses blood pressure and spirometry. But a lot of the labs that are in these two books have been incorporated or put into the Project Lead the Way curriculum. So even though the Project Lead the Way curriculum, which is a specific curriculum for the biomedical courses, a lot of the experiments and instructions are actually taken from our books, and then they add a lot more material. So it would be worthwhile getting these books if you wanted them, because you could just use them on your own and then compare how we've set it up to how Project Lead the Way has set them up. But we work really well with Project Lead the Way, talk with them. There's a whole series of videos that are in the, there's a whole series of videos on how to use these sensors that Project Lead the Way has in their, where you log in and you can look at their curriculum and everything. So you might see pictures of me, or at least hear my voice showing you how to use these sensors there as well. All right, so let's just talk about EKGs. That's what a lot of people want to do, is they want to measure EKGs, or record the electrical activity of the heart. And they're really interested in seeing something very particular, and that is the different waveforms in the EKG. Don't worry, the presentation that I'm going through, you do have access to, and you can get access to that as well. And Dylan can show you. It should be, yeah, she just dropped the presentation. It's part of the Google slide deck that's there. The one thing to be aware of is that all of these sensors, the Go Direct ones, do not require an interface. So here is the Go Direct EKG sensor right here. And it does have a Bluetooth. So if I tap on this button, I can connect to it wirelessly. But really importantly, they all have a USB port. So the simplest way to get them to work is always going to be using them over USB. And that's just what I'm going to show you now. So there's my Go Direct EKG. I've got some leads right here. I'm going to connect some electrodes to myself. One to my left arm. One to my right arm. One to my wrist. Our instructions or other instructions will tell you to put them higher up. I can't show you that though if they're on my biceps, but they work fine here. But you could put them farther up the arm. The higher up you place these electrodes, the better signal you're going to get. It's always just going to be simpler to connect to USB, and I'll explain that to you or show that to you here in a minute why. So let me go to graphical analysis. So what I'm going to do next is I'm just going to show you how to set up and compare and measure like my EKGs. And really what we want to see are these waveforms, the P, the QRS, and T waveforms. So let me launch graphical analysis. What's great is if you launch graphical analysis and you just plug in the Go Direct EKG over USB, it just automatically sets itself up. So it just says, oh, hey, you have an EKG sensor. I think you probably want to sample at 200 samples a second. And I know that the standard human physiology lab is 3 seconds long. So there's no other steps that you have to worry about. All you have to do is worry about connecting the leads. So I'm going to take the leads right here, connect the right lead to my left arm, green lead to my right arm, and black to the reference, which is just on my wrist. And then I'm going to sit quietly for a minute, and then I can't talk. I'm going to hit collect. There we go. Now I'm going to unconnect myself. And you should be able to see that that's a really nice EKG trace. There's the P wave, QRS, and the T. And my heart rate's a little fast because I'm giving a presentation in front of a bunch of people that I don't know or haven't met. And you can also like go across here and you can take a look and see the time differential. if you wanted to look at different segments by looking at view statistics. That'll give you the delta X, which would be the time. Someone brought up a really good question, which is, what about interference? That's a really great point. That is a good reason to use USB if you want to. So let me disconnect, and I'll show you how to connect over USB. And just remember that the way that I'm connecting wirelessly over Bluetooth, this is the same for all Go Direct sensors. So like right now, if you can see this little Bluetooth icon, this little is flashing, that means it's broadcasting. I'm going to hit File, New Experiment. New experiment is always your friend. It sets everything up. And you can see now it's like, hey, I don't see anything connected, right? I don't see anything there, but you will see that it says GDX EKG, and then it has these numbers, G4. So let's take a look back at the camera, and we should be able to see, yep, it says G4 right there. So that's the right one. Let me zoom back out. All right, let me connect myself. I see another great question. I promise I'll get to that one too. I'm just going to sit quietly. I'm going to connect to the EKG sensor. So if you're using Bluetooth, you have to choose to connect. So if you're on iOS or Android or something like that, you would connect to that way. Oh, come on there. Let's see. Oh, it went to sleep. Hold on here. Let me turn it back on. There we go. And now let me connect, collect an EKG trace. So you can see the signal is a little bit cleaner, but looks very similar. So it's really up to you. The simplest way though, if you're on a Chromebook, is to actually use, if you're on a Chromebook, is to use USB. Everything's going to be smoother. Everything's going to work fine or work better. All right. So someone had a great question, like what if you have multiple Bluetooths, like multiple EKG sensors or other sensors or 8 groups running the lab at once, all near one another. That's what's great about Bluetooth and or USB. They don't interfere with each other, right? So if you plug them in over USB, they're not going to interfere with each other. If you're using Bluetooth, Bluetooth is peer-to-peer. It isn't broadcasting everywhere, so it's not going to interfere. The one key thing is to make sure that students recognize or know what that sensor is, know the name. There is a way to identify it. Let me see if I can show you that here. So if I go back to the EKG sensor here, I can click on identify, and then if I hit identify, see how it's flashing right there? Then let me show you, it's actually also flashing right here. So you can hit identify in GA. Oh, oops, hold on. If you hit identify in GA, it'll show you that, like how it'll show you which one is there. So you can make sure that they're connected to the right one. All right, will the older EKG, yes, the older EKG sensors will work, and that's what I'm gonna show you next. Great. How do you adjust the 0 and the x-axis to line up the EKG? Well, the 0 axis doesn't really matter in this case. You could 0 it, but it's going to cycle around it. If you wanted to adjust the baseline, there's ways to do that. You could just add a little bit to the baseline. Oh, no worries, Allison. You can keep on bothering me. Let's see here. What other questions do we have? Okay, hold on there. For Bluetooth, having students shut off their Bluetooth, that won't really be a problem if you're trying to connect. It may help, but it may not. But like I said, Bluetooth is peer-to-peer. So what's great about the Go Direct sensors is you don't have all of the same noise issues that you normally would have using the old sensors. How do you save? Don't worry. I'm going to show you how to save this file and all that too. Great question. To save this file, you just go to the top left-hand corner and you can go to Save As. And you can just give it a name. Another really cool thing that I wanted to show you is, let me just show you this. I'm going to hit File, New Experiment, don't save. You could make up your own, what we call experiment files. So I could say, hey, I want this recording to last for 10 seconds. And then I could hit Save As. And I'm going to call this, let's see, desktop. There we go. I'm going to call this EKG 10 seconds. Great. And then I'm just going to close this. And let me relaunch graphical analysis. Great. Okay, so great. I'll repeat the question since everyone's not broadcasting them to everyone. So I'll make sure that I repeat the question that's being asked. Thank you so much. Now, if I have an experiment file that I've created, I can go up here to where it says Open Save File, and I can say Choose File. And I can actually open up that EKG 10-second experiment file, open it up. And let's see, has it grabbed my EKG trace? I'm going to have to turn on the EKG sensor, connect to it. Or connect to it first, and then I can go up here to File, Open, EKG 10 seconds, and open, and everything's already set. So you can, as long as you're connected, you can open up a file, you can open up a setup file, and the experiment will be set up for you if you want. Okay, so someone else asked, can you share these saved experiments? Oh yeah, you can save saved experiments, you can share data, you can do all of that by a couple of ways. You can either just save it to a Google Drive and email it, move it over. We also have a great way to do data sharing. Data sharing means I can send that data out to everyone that's here. But before we do that, let's move on a little bit, and if we have time at the end, I can talk to you about that. But yeah, you can easily save it and share files. You can print. The best way to really print is to export. So you can export the data as well as a graph, a CSV, an AMBL, or a PDF. So there's lots of things that you can do. All right, so quick. Someone asked about the old EKG, right? So let's talk about the old EKG sensor. So you can do the same thing with graphical analysis with the old EKG sensor. So here is, oh wait, hold on, I don't want to do that. Sorry. Give me 1 minute there, just opened up the wrong experiment. Here, let me launch over here, show you here. Here's the old EKG sensor. It's got an order code EKG BTA. That's because it has this connector here on it. And then you need to use the older sensors with an interface. Here's the new LabQuest Mini. Here is an older LabQuest Mini, but you need to use an interface. The interface has 3 analog channels, 2 digital ones. So you plug it into, I'm going to plug it into channel 1 here. And then I need to make sure that it's plugged into USB. So I have a nice USB cable here. I'm going to plug it into the back of it here. And then I'm going to go and launch graphical analysis. So let me move away from here. Great. And hopefully what you all can see is graphical analysis has seen that there is an EKG sensor attached. It's already set up the same data rate. I'm in the same version of graphical analysis. Let me change the app preferences so you can see it a little better there. Right, so everything's set up, everything's ready to go. I'm just gonna connect myself to the leads again. This time I'm using the older EKG sensor. I'm gonna sit still. I'm gonna use all my old tricks, like hold it in my hand and stuff, and let's see how the EKG trace looks. There, looks pretty nice. My heart rate's going a little high, but that's a decent EKG trace. Right, there's P, QRS, and T waves. So in this case, once again, just to show you that this is real, this was all done with the older EKG sensors, right? And this is done in graphical analysis. And you could easily do this with a Chromebook or a Mac or PC. Where this would be tricky with your old sensors is if you're trying to use it on a tablet or a phone, because those don't have USB ports. So that would be tricky. But as far as Chromebooks, you can use your LabQuest Mini, your old sensors, and use them use a lot of your physiology equipment with them, just like I did right here. So questions on that? And really, it should be as simple as plug and play. You just plug the LabQuest Mini in to your Chromebook or your device, plug in the EKG sensor, follow instructions, and get going. And if you have any questions whatsoever on how to do that, or if you want to modify instructions, because the books that we have, the Human Physiology Experiments book, they're actually written for Go Direct sensors and for graphical analysis. But it's super simple to modify most of the instructions. So if you want or need help modifying any of those instructions, just let us know. If you buy those books, you actually get access to them. So I'm logged into my account, and you can actually then create an account once you've bought the book. You can download the experiments. And once you've downloaded those experiments, we have all of the experiment instructions in Word and PDF format, and you can easily modify them and change them. All right, so for Go Direct sensors, they work great. There's lots of special things about them. They work on anything. For your older sensors, you're gonna have to have something that has a USB port. Chromebook works fine, Mac and PC work fine. Not gonna work if you have iOS devices, phones, tablets, Android devices that are, tablets or phones either. All right, so. No, that's a really good thing is there are no what we call experiment files for graphical analysis. We got away from that in Logger Pro because it's just really hard to set up, but be super easy to make those yourself or to talk to me and I can help you make them, what we call the setup files. In some cases, like for the EKG experiment, there's no modifications whatsoever. For others, you would need to modify them. So someone asked, like, does graphical analysis have the saved files for the experiments in the books? For a lot of the old books, Biology with Vernier, Chemistry with Vernier, Human Physiology with Vernier, we had experiment files for Logger Pro. And what we found is those are hard to push out across all platforms. So we have not made those available for graphical analysis just because it could be really hard to make someone's Android tablet do special things that you might be able to do on a Mac. So we don't do that. But if you want to create them yourself, setup files, just get in touch at biology@vernier.com if you have old or new sensors, and I can help you make them, show you how to go through making them. And I can even tell you, like for a lot of the EKG experiments, you don't need to. Everything's already there. You just plug in and go. But that leads us to a great question, which is what about EMGs, right? So, and by the way, if you want any of the experiment files that I'm showing you here, just email biology@vernier and say you want them, and I'll give them to you. That won't be a problem. I don't know if you can use Google Classroom, but what I'm using for graphical analysis, but I'm sure that you could share files, put them in there, and move them back and forth. So someone asked, can you use Google Classroom for doing EKGs? What I'm running is graphical analysis, which has to run on a device. If you might be able to launch the PWA in Google Classroom. and run it there, but I don't have access to Google Classroom, so I can't give you the perfect answer. So what about EMGs? All right, so one thing to show you is that EMGs, electromyographs, electrical activity of muscles, you use special filtering. And once again, you can create a special file, but also there are special channels that exist on the Go Direct EKG. So let me give you an example of that. So what we would want is to see data that looks like this. This is the electrical activity of a muscle, the forearm, and then I've rectified it. So I've made everything that's negative here positive. And I also could create setup files as well to get all this. So let me just give you an example here of what I'm talking about. So let's go back to graphical analysis. I'm going to disconnect the LabQuest Mini. I'm going to select File New because File New is my friend. And I'm going to plug in the EKG sensor. I mean, the Go Direct EKG sensor. So just say here, File New, don't save. Now, the default channel that it goes to is EKG, but if I go down here in the bottom right corner and I select Sensor Setup, one of the great things about Go Direct sensors is they have sensor channels. So that means I can turn on other channels. Lots of Go Direct sensors have other channels that like have temperature attached. This one, we have special other channels so that you can look at heart rate or EMGs or EMG rectified, or if you wanted to, you could look at it without any filtering for testing it. I'm going to turn on these two. This just changes the filtering. If you want to look at the electrical activity of a muscle, you need to use different types of digital or electronic filters to look at them. There's a more high frequency signal. And so now what I'm going to do is I'm just going to, let me go back to the camera. I'm going to attach an electrode here to my forearm. So you can see there on my forearm. And then I'm going to be looking at muscle activity across here, these 2 points, and I'm going to be putting the ground here. So let me just do that really quick. Here is red. Here is green. Black is reference. Oh, that's coming loose. Grab another electrode here. Let's put a black reference. All right, I'm gonna minimize this again. I'm gonna change the rate here a little bit. I don't want to record this fast. I'm just gonna record for 100 seconds, and I just gonna record for 10 seconds. And then what I'm gonna do is I'm gonna squeeze on something while I'm doing this. So I'm just gonna squeeze. Here's a hand dynamometer. I'm not gonna plug it in right now, but I'm just gonna squeeze on it to create some electrical activity in my muscle. And then I'm going to use my left hand. Oh, here we go. So I'm going to hit collect, and then I'm going to sit for 2 seconds and then I'll squeeze a little bit and then squeeze harder and squeeze harder. Okay, collect. Squeeze. 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 There we go. And now I'm going to hit zoom in. That's this button here, zoom into all data. So you can see I squeezed a little bit and there's a little bit of electrical activity. I squeeze harder, there's much more electrical activity. And I squeeze really hard and there's even more. And if I tap on the y-axis, I can choose which one of these I want to see. The EMG is both positive and negative deflections. I can also just make all of those positive by hitting EMG rectified. And that's really nice because it's just easier to analyze because I could actually select a range and then get the integral. Now, like I said, though, what's great about graphical analysis is you could create your own setup file for this as well. So for example, I could go to new experiment, don't save. And I could say choose file. and I could pick up EMGs setup. And now what you should see is it's already set up. So it's already set up to measure EMGs rectified. It's set to measure for 20 seconds instead of 10, and everything's all set up. So this is just a setup file. And if I hit collect, so I didn't have to go through all those other steps of selecting sensor channels. And there we go. I can squeeze a little bit, relax. Squeeze a little bit more, relax. Squeeze, relax. All right, so let me answer a few questions here. All right, so someone asked, this is a really good question. So am I correct in understanding that the interface sensors, either mini or the big green one, this doesn't work as well? Okay, two things there. The Lab Pro is not supported in graphical analysis, so you won't be able to use the Lab Pro. That's the green thing that looks like a calculator. I love those things. I use those things when I was teaching. Those still work in Logger Pro. They won't be able to work with graphical analysis on a Chromebook. The LabQuest Mini works fine on a Chromebook. The LabQuest Mini and your analog sensors will work fine. They don't work as well, but I can help you get them to work. It's just you're going to have to work a little bit more to get them to work well, but like the EKG sensor will work. You can use the EKG sensor to do EMGs too. They will work with graphical analysis, so you don't have to get rid of them. The best thing to do is get in touch with me or my team, the biology team at Vernier, and we can help you get it so that you're comfortable using them on a Chromebook or a Mac or PC in graphical analysis. If you don't have access to Logger Pro and you have the BTA sensors, the older ones, and an interface, you still can use them. Oh, LabQuest 2 will work fine with Go Direct sensors. So someone asked, what about LabQuest 2? People also ask, what about LabQuest 3? Those will work with Go Direct sensors and old ones. It just depends on how you want to use them or how you're trying to use them. And then don't forget, a LabQuest 2 can be used as an interface with a Chromebook. You can just plug a USB cable into it. All right, hold on here. You guys are, all of you are asking wonderful questions. So someone asked, how would I use statistics? Well, statistics is actually really great if you select a region and then you tap down here under graph options. I mean, you can apply curve fits, you can do an integral, you can also view statistics. And under statistics, it's going to give you a couple things. It's going to give you the range that you selected, so the X range, which will be in seconds. It's also going to give you the peak amplitude, the delta Y, which is 7 millivolts. It'll give you the mean and the standard deviation, and then your minimum value and maximum value. So it can be a really useful way to go through and take a look at any signals that you've got. You also can just tap and analyze. So you could just tap right here and say, okay, well, that's 5.5 seconds. Go to the end, and that's 8.23, and then subtract to find out what the duration is, or just find out right there, oh, the peak is 0.71, right? So you can also just inspect by putting this marker here and grabbing along and taking a look at the data. All right. You're asking so much questions, so many great questions. Now I can do the same thing with the Vernier EKG sensor. I could set that up and I could record EMGs with it too. There's no problem doing that as well. I could even set up special filtering to make it work better, but you don't have to. But once again, you could just set it up. I could show you that real quick if you're interested. So let me disconnect from the LabQuest Mini. From the Go Direct EKG, say new experiment, don't save. I'm gonna connect to the LabQuest Mini. I'm now gonna plug in the EKG sensor is already connected. Great. So the EKG sensor is already connected. I'm just gonna go here and I'm gonna say, hey, just record for 100 at 100 samples a second, record for 10 seconds. And then all that I'm gonna do is I'm gonna attach the same EKG sensor leads to my arm. And then I'll just do the same thing here. I'll hit collect and we'll record some data. Squeeze. Squeeze really hard. Squeeze. And then I'll hit zoom in. So you can see that you can also record EMGs as well. Now, what's nice with the EKG sensor is there's another channel here called potential that isn't filtered. I can turn that on and you can see that it looks much more like an EMG trace. So if you want to, you can turn filtering off and do that, and that should work. That should work great, and you'll get much better EMG signals. There's other ways to process it. The most important thing for you to understand is your older sensors, the EKG BTA or the EKG sensor, still can be used to record EMGs and EKGs. You should be fine. All right, so someone asked, this is a great question. What about the Go Direct heart rate monitors? That's a whole nother thing. I don't think that we have time to really talk about those because I've only got like 5 minutes left. But if you have questions, like I said, about anything, all that you need to do is contact us at biology@vernier.com and I'll take you through it. The way that if you're having problems connecting a Go Direct heart rate on graphical analysis, the best thing to do is to pull out the module. Let me just show you what to do really quick, because this will help if you can't get it to work or you can't get it to connect. Let me just go back to the camera really quick here. Move this out of the way. If you have the Go Direct heart rate, it may have either a polar module. So this is the polar H9 module, or it may have a module that looks like this. This is the polar module. These are the ones that support Bluetooth. And what you can do is you can flip them over and you can see that they actually have these places where the batteries are. And what I would recommend is removing these, taking out the battery. Once the battery's removed, just put your fingers on these leads and hold it there for 10 seconds. That resets the module, and then put the battery back in. That fixes about 90% of the problems that I've seen with both of those sensors. All right. Are there other questions that folks have? Let me get out of here for a minute. Get out of here. Oh, the license key. The license key, we can get you the license key for GA Pro. Hold on. Someone asked for the license key again. Let me hold on and get that for, I'll get you for that here at the end. Are there any other questions? I was just going to show you, like, once again, you can use the EKG sensor with the hand dynamometer at the same time. And you know what you can also do? You can also use the EKG sensor with the Go Direct gas pressure sensor. If you're a project lead the way teacher, what a lot of people are using instead of the hand dynamometer is they're actually using a little pressure sensor bulb. So we have a bulb. You can attach this to a gas pressure sensor or a Go Direct gas pressure sensor, and you can make it work like a hand dynamometer. So if you have a gas pressure sensor floating around, a Go Direct one or another one, you can just buy a little bulb to turn it into something like a hand dynamometer. And then just once again, all of your old stuff still is really useful. So this is an older hand dynamometer, a gas pressure sensor, and an EKG sensor. You can still use all of that equipment. So most of your older sensors, human physiology sensors, all still can be used with graphical analysis. All right. So someone asked, since you do not have experiments loaded, do you have to connect the sensors before setting up your own experiment? Yes, if you want to create a setup file, what you need to do is just connect the sensors, make sure they're connected, set everything up that you want, just don't collect data, and then just save it. That's basically a setup file. And then you can reopen it and use it, and it should work fine. All right. If you have some time, please fill out the survey that Dylan left. Hopefully you all found that this was useful. If you want us to do another webinar like this, I can cover other sensors. Don't forget that we also on our website, let's see here, we have a whole section for educator training, and we have past webinars there as well. And I have done webinars on lots of different things. We've done webinars on the spirometer. We've done webinars on other things, on spectroscopy. Just go ahead and take a look through here, and you're probably going to find something really useful. We also have our own YouTube channel, and the YouTube channel has lots of really good tips and tricks on how to use sensors. So thank you so much for coming. I really appreciated having you here, and that's really all the time we have.
Thanks, everybody. Have a great rest of your day.